Hello everyone, this is Vaibhav Shandilya, MBBS student from ESIC Government Medical College, Hyderabad, and today we are going to talk about a fungus disease which affects the bloodstream. It's caused by Candida albicans and it's known as Candidiasis. Okay, so let's get started. We're going to start with the introduction as usual. So Candidiasis is the most common fungal disease in humans, fungal. And characteristically, Candida albicans, it produces pseudohyphae. This is very important to remember, whether it's producing a hyphae or a pseudohyphae. So here, pseudohyphae. Coming to the pathogenesis, so let's talk about the predisposing factors first. So the predisposing factors we have are extremes of age. Remember, it shows a bimodal expression. Bimodal meaning it shows like in infancy and elderly. These are the two age groups which will show this disease. So extremes of age, pregnancy, then broad spectrum antibiotic use, transplant surgery or immunosuppression or HIV, diabetes, iron deficiency, zinc deficiency. All these, make the immune system compromised of the patient, making them susceptible to candidiasis. So these are the main predisposing factors. Coming to virulence factors, or the main part of the pathogenesis, firstly, adhesion is through adhesins, through which it attaches to the mucosal surfaces. Then enzymes for invasion are aspartate, proteases, serine proteases, right? And toxins used for tissue damage are produced from the Candida cell wall and are pyrogenic in nature. They cause fever in the individual. And further, the pseudohyphae, which is formed by the Candida albicans, it indicates an active Candidiasis infection. Active Candida infection. The pseudohyphae. Next, adaptation. How does the Candida albicans adapt? It regulates in three different forms. It transforms in the hyphae, pseudohyphae, and yeast form, and this is how it escapes from the host immune system by adapting in these three different phases as per its requirements. This is known as phenotypic adaptation. Okay, phenotypic adaptation. Right, so this is the virulence factors. We have adhesins, these enzymes, aspartate, serine proteases, toxin, pyrogenic, which is produced from the cell wall of the Candida, pseudohyphae indicating active infection, and adaptation in three forms: hyphae, pseudohyphae, and yeast form. Okay, let's go to the next aspect, which is the clinical features and manifestations. So we have four major manifestations. Firstly, we have the invasive candidiasis, then mucosal candidiasis, then cutaneous candidiasis, and finally allergic candidiasis. Right, so let's start with invasive candidiasis, which basically expresses as hematogenous spread. So when Candida spreads through hematogenous route, there occurs invasive candidiasis. And this hematogenous spread is in different parts like the pulmonary system. It can cause UTIs. It can cause ocular manifestations like endophthalmitis or keratoconjunctivitis. It can go in the joints, cause arthritis, go in the brain and causes meningitis, go in the bones, osteomyelitis, and in the diffused in the bloodstream causes septicemia. Right. So it basically covers almost all systems: pulmonary, urinary, then ophthalmological, joints, brain, bones, and the bloodstream. Right. So this was invasive candidiasis. Next, talking about mucosal candidiasis, it manifests in two different diseases, which is basically vulvovaginitis and oropharyngeal candidiasis. This will be covered in the respective, like this vulvovaginitis is a different topic under the urogenital section, but namely mucosal candidiasis involves vulvovaginitis and oropharyngeal candidiasis. Then cutaneous candidiasis means something to do with the skin. So we have intertrigo, which are basically these pustules on the skin folds, right? So intertrigo, and then we have paronychia and onychomycosis, which are basically nail bed infections. Okay, these are nail bed infections: paronychia and onychomycosis. So these are the cutaneous candidiasis. Finally, allergic candidiasis, which occurs because of hypersensitive reaction and exaggerated immune response to the Candida antigens. This is known as a candidid reaction. Okay. This is found in many different organisms. For example, the hydrated cysts show hydrated reaction. Similarly, here the Candida antigens show candidate reactions. Right. So these are the four forms: invasive, mucosal, cutaneous, and allergic candidiasis. This is the clinical manifestation. Coming to the lab diagnosis, very important part of microbiology. The specimen taken is from the blood and urine. Direct examination, we see gram positive oval budding yeast cells with pseudohyphae. Characteristic statement you need to remember. This is of most importance. So these are the oval budding yeast cells, and you can see that they have pseudohyphae, right? These are all pseudohyphae. And so these are gram-positive oval budding yeast cell with pseudohyphae. You need to remember that. Culture in Severod's texture shows cream white pasty colonies. This is also very much characteristics. Everything in the lab diagnosis must be remembered. This is a very important organism. Cream white pasty colonies seen in STA agar, and this is how they look under the STA, cream white pasty. And next, coming to the identification, there are multiple types of tests done for cultures done for identification. We have the germ tube test, which is shown by the Candida albicans. As you can see, we see that there is a formation of a germ tube here, right? This is germ tube formation, characteristic for Candida albicans. We have Delmauch plate culture. We have growth on chrome agar. Chrome means color, so we can see this characteristic growth on chrome agar, and this light green color is for Candida albicans, right? This you can see the arrow pointing. So this is growth on chrome agar. Next we have growth at 45 degrees Celsius. This is also positive characteristic for Candida albicans. And we have carbohydrate assimilation and fermentation test. So basically we have in identification, we have germ tube test, Delmauch plate culture, growth on chrome agar, growth at 45 degrees Celsius, and carbohydrate assimilation and fermentation test. Earlier in culture, we showed creamy white pasty colonies, and direct examination, gram-positive oval budding yeast cells with pseudohyphae. Coming to the automated techniques, we have Maltitov-Vitek. We all know that. Molecular, we have PCR. Antigen detection is by cell wall Manan antigen. This is characteristic. Cell wall Manan antigen. Antibody detection is by anti-cell wall Manan antibody. Okay, just easy. Next, we have enzyme detection using enolase. There is enolase positivity. That's how you detect enzyme. Then we do a beta-D glucan assay, which is basically a marker of this invasive fungus, which is Candida albicans. Beta-D glucan assay. Okay, so this is the lab diagnosis broadly. Specimen, direct exam, culture, identification, automatic methods, molecular, antigen detection, antibody detection, enzyme detection, and beta-D glucan assay. Okay, so this was Candida. Let's go over it briefly. The Candida albicans produces pseudohyphae. Pathogenesis, predisposing factors are which compromise the immune system. For example, pregnancy, extremes of age, antibiotic use, transplant immunosuppression, HIV, diabetes. Virulence factor, we have adhescents, aspartate serine proteases, toxins, pyrogenic toxins. Then there is a phenotypic adaptation in 3 forms: hyphae, pseudohyphae, and yeast. Clinical manifestation 4 forms: invasive candidiasis, mucosal candidiasis, cutaneous candidiasis, allergic candidiasis. And in lab diagnosis, as discussed, we have the discussed points. So it is very easy, Candida albicans, but it is a very important question. You are sure to get this in your university examinations one time or the other, definitely. Okay, so hope you liked the videos. Please support this channel. Thank you.